Greetings to everyone. I'm Freddy, and together with Jul Errol, John Raymond, Ferry Ann, Lord William, Maria Lace, and Chari, we're going to discuss the steps, what, and how the citric acid cycle works. Now, let us hear from Maria Lace Retita for the introduction.
So this metabolic pathway we will be reporting is called by three different names. The first one is citric acid cycle. This name refers to the first molecule that forms during the cycle's reaction, the citrate, or in its protonated form, citric acid. You may also hear this series of reaction called tricarbocyclic acid or the TCA cycle because of the three carboxyl groups on its first two intermediates. And lastly, the name Krebs cycle is named after its discoverer, Hans Krebs, the German-born British biochemist Hans Krebs, who pioneered work on the chemical processes involved in metabolism. While working in Germany, he discovered urea, the key chemical process involved in excretion. Following the rise of Nazis in 1933, Krebs moved to Britain where he continued his research. Then by 1937, through experimenting on the pigeon flight mussel, he discovered the process by which body cells convert food into energy, a process now known as the Krebs cycle. For this, his greatest discovery, Krebs shared the Nobel Prize for Physiology or Medicine in 1953 with the US biochemist Fritz Lipman. So this cycle is the main source of energy for cells and an important part of aerobic respiration. Citric acid cycle is the key metabolic pathway that connects carbohydrate, fat, and protein metabolism, which is important in all the living cells that use oxygen as part of their cellular respiration. For the next slide, as you can see in the picture, the molecules citrate and isocitrate are the first two intermediates of the citric acid cycle. So why is citrate converted into isocitrate? So because the tertiary hydroxyl group is not properly located in the citrate molecule for the oxidative decarboxylation in the process. So citrate is then converted into isocitrate by the enzyme aconitase. This is accomplished by the removal and addition of water to yield an isomer. So this process will be further discussed by the next reporter. And when citrate gains 3 hydrogen ions so that it has no longer negative charge, then it will be called citric acid. The citric cycle is the central driver for cellular respiration. Cellular respiration is the metabolic pathway that breaks down glucose and produces ATP. So it takes acetyl coenzyme A which is produced by the oxidation of pyruvate and originally derived from glucose as its starting material, and in a series of redox reduction harvest much of its bond energy in the form of NADH, FADH2, and ATP molecules. The reduced electron carriers NADH and FADH2 generated in the TCA cycle will pass their electrons into the electron transport chain and through oxidative phosphorylation will generate most of the ATP produced in the cellular respiration. So basically, in the mitochondrial matrix, which by the way is where the TCA cycle occurs, the enzymes of the citric acid cycle produce the reducing equivalents of NADH and FADH2. They deliver electrons to the complexes of mitochondrial electron transport chain, which builds up a proton gradient that drives ATP production. So this signifies that the Krebs cycle is actually a significant part of the much larger process called cellular respiration, the process where our body harvests energy from the food. So that's all for the introduction. Thank you.
The citric acid cycle, like the conversion of pyruvate to acetyl coenzyme A, occurs in the matrix of the mitochondria in the karyotes. Both of these stages occur in the cytoplasm of prokaryotes. The citric acid cycle is a closed loop in which the molecule used in the first stage is reformatted in the last phase. There are 8 primary steps in the cycle. In the first phase of the cycle, acetyl CoA combines with oxaloacetate, a 4-carbon acceptor molecule, to produce citrate, a 6-carbon molecule. Following a rapid rearrangement, the 6-carbon molecule releases 2 of its carbons, as carbon dioxide molecules in two separate processes, each yielding a molecule of NADH. The enzymes that catalyze these processes are important regulators of the citric acid cycle, speeding up or slowing it down, depending on the energy needs of the cell.
Good day, this is Ferandi Pabala, and to continue the report about the overview of the citric acid cycle. So, to continue the report about the overview of citric acid cycle, we are now down to the 4 remaining carbon molecule of Krebs cycle or the citric acid cycle. So, these 4 carbon molecule will go through a series of reaction. First is to convert the guanosine diphosphate or JDP to JTP, the guanosine triphosphate. So JTP is similar as ATP, adenosine triphosphate. Adenosine triphosphate is principal energy carrier in the cell, while JTP or guanosine triphosphate has a specific rules in many signaling pathways. So the next step is to reducing the electron carrier FAD or flavin adenine dinucleotide to FADH2, the dihydroflavin adenine dinucleotide. So and the last is to produce another NADH or nicotinamide adenine dinucleotide. And this reaction creates molecules that can repeat the cycle. So in overall, the citric acid cycle releases 2 carbon dioxide molecules, 3 NADH, 1 FADH2, and 1 JTP. So citric acid cycle happens twice since glucose that enters cellular respiration consists of 6 carbons, and these 6 carbons will be divided into 2 pyruvates, and 1 pyruvate will consist 3 carbons. So and every glucose there is 2 acetyl-CoAs that was made, and this glucose will consist 2 acetyl-CoAs.
Hello there. Once again, I'm Freddy, and at this juncture, we're going to discuss the steps of the citric acid cycle. Before we start, let us have a brief overview. How important is the citric acid cycle? So important that it has not 1, not 2, but 3 different names in common usage today. The citric acid cycle, as we will use here, is named after the first molecule formed throughout the cycle's reactions, citrate, or in its protonated version, citric acid. This set of events is also known as the tricarboxylic acid or TCA cycle, named after the 3 carboxyl groups found in the first 2 intermediates, or the Krebs cycle, named for its discoverer Hans Krebs. Now let's go to the 8 steps of the citric acid cycle. Step 1. In the first step of the citric acid cycle, acetyl-CoA interacts with oxaloacetate, a 4-carbon molecule, to release the CoA group and synthesize citrate, a 6-carbon molecule. Step 2. Citrate is transformed to its isomer isocitrate in the second step. Because this is a 2-step process that involves first removing and then adding a water molecule, the citric acid cycle is frequently referred to as having 9 steps instead of the 8 that we are featuring now. Step 3. Isocitrate is oxidized in the third step, releasing a molecule of carbon dioxide and leaving behind a 5-carbon molecule called ketoglutarate. NAD positive is converted to NADH during this phase. Isocitrate dehydrogenase, the enzyme that catalyzes this step, is crucial for controlling the citric acid's cycle phase.
Let's now proceed to the 4th steps of citric acid cycle. The 4th step is similar to the 3rd. In this case, it's a ketoglutarate that's oxidized, reducing NAD positive to NADH and releasing a molecule of carbon dioxide in the process. The remaining 4 carbon molecules picks up coenzyme forming the unstable compound succinyl. The enzyme catalyzing these steps, ketoglutarate dehydrogenase, is also important in regulation of citric acid cycle. In step 5, the acetyl of succinyl is replaced by a phosphate group, which is transferred into ADP to make ATP. In some cells, GDP or guanosine diphosphate is used instead of ADP, forming GDP or guanosine triphosphate as a product. The 4 carbon molecule produced in this step is called succinate. It means that a CO2 molecule is released and that an oxidation takes place. In this case, that a carbon atom that will form into CO2 is oxidized. Furthermore, an energy-rich coenzyme molecule is added to a succinyl is formed. At the end of this reaction, we get a C4 body. That the GTP is similar to ATP. Both serve as energy sources, and the two can be readily converted. In which of the two molecules is being produced during the citric acid cycle depend on the organism of the cell type. For example, ATP is made from the human heart cells, but a GTP is made in the liver cells.
So now we will proceed to the next step in the citric acid cycle, which is the step 6. Step 6 is a dehydration process that converts succinate into fumarate. 2 hydrogen atoms are transferred to FAD, producing FADH2. The energy contained in the electrons of these atoms is insufficient to reduce NADH, NAD+, but adequate to reduce FAD. Unlike NADH, this carrier remains attached to the enzyme and transfers electrons to the electron transport chain directly. This process is made possible by the localization of the enzyme catalyzing in the inner membrane of the mitochondria. Always remember that FAD is a better acceptor than NAD+, meaning it has a higher affinity or hunger for electrons. Succinate is not a great electron donor. It means that it has a fairly high affinity for electron itself and is not eager to give them up. NAD+ is not electron hunger. It means it is enough to pull the electrons away from succinate, but FAD is. The next step in the citric acid cycle is step 7. In step 7, water is added to fumarate to produce malate. Water is added to 4 carbon molecule fumarate to produce another 4 carbon molecule malate. The next step is step 8. It is the last step in the citric acid cycle that regenerate oxal acetate by oxidizing malate, and another molecule of NADH is produced. The starting 4 carbon compound is regenerated by oxidation of malate, and then another molecule is produced of NADH+ is reduced to NADH in this process.
Products of the citric acid cycle. In a single turn of the cycle, 2 carbons enter from acetyl coenzyme and 2 molecules of carbon dioxide are released. 3 molecules of nicotinamide adenine dinucleotide and 1 molecule of flavin adenine dinucleotide are generated. 1 molecule of adenosine triphosphate or guanosine 5-triphosphate is produced. These figures are for 1 turn of the cycle, corresponding to 1 molecule of acetyl coenzyme. Each glucose produces 2 acetyl coenzyme molecules, so we need to multiply those numbers by 2 if we want the pure glucose yield. 2 carbons from acetyl coenzyme enter the citric acid cycle in each turn, and 2 carbon dioxide molecules are released. However, the carbon dioxide molecules don't actually contain carbon atoms from the acetyl coenzyme that just entered the citric acid cycle. Instead, the carbons from acetyl coenzyme are initially incorporated into the intermediates of the cycle and are released as carbon dioxide only during turns. After enough turns, all of the carbon atoms from the acetyl group of acetyl coenzyme will be released as carbon dioxide.
And so, the question is, where's all the ATP? So, it is true that the citric acid cycle does not produce much more of ATP. But with the help of NADH and FADH2 cellular respiration, these electron carriers will connect to the last portion of cellular respiration and will deposit their electron into the electron transport chain that will drive the synthesis of ATP molecules through oxidative phosphorylation. So oxidative phosphorylation is a highly efficient method of producing large amounts of ATP. So it is where all the ATP was being produced. Oxidative phosphorylation.
We would like to end our presentation with the key points and key terms that we learned in this discussion. Key points. The 4-carbon molecule, oxaloacetate, that began the cycle is regenerated after 8 steps of the citric acid cycle. The 8 steps of the citric acid cycle are a series of redox, dehydration, hydration, and decarboxylation of reactions. Each turn of the cycle forms 1 GTP or ATP as well as 3 NADH molecules and 1 FADH2 molecule, which will be used in further steps of cellular respiration to produce ATP for the cell. In the conversion of pyruvate to acetyl-CoA, each pyruvate molecule loses 1 carbon atom with the release of carbon dioxide. During the breakdown of pyruvate, electrons are transferred to NAD positive to produce NADH, which will be used by the cell to produce ATP. In the final step of the breakdown of pyruvate, an acetyl group is transferred to coenzyme A to produce acetyl-CoA. The Krebs cycle is a series of chemical reactions used by all aerobic organisms to generate energy through the oxidation of acetate, derived from carbohydrates, fats, and proteins, into carbon dioxide. Theoretically, there are several alternatives to the TCA cycle, but the TCA cycle appears to be the most efficient. The citric acid cycle consumes acetate in the form of acetyl-CoA and water, reduces NAD positive to NADH, and produces carbon dioxide. Now let's go to the key terms. Citric acid cycle, a series of chemical reactions used by all aerobic organisms to generate energy through the oxidation of acetate derived from carbohydrates, fats, proteins into carbon dioxide. Krebs cycle, a series of enzymatic reactions that occurs in all aerobic organisms. It involves the oxidative metabolism of acetyl units and serves as the main source of cellular energy. Mitochondria. In cell biology, a mitochondrion, plural mitochondria, is a membrane-enclosed organelle, often described as cellular power plants, because they generate most of the ATP. Acetyl-CoA, a molecule that conveys the carbon atoms from glycolysis pyruvate to the citric acid cycle to be oxidized for energy production. Acetyl-CoA. Acetyl coenzyme A or acetyl-CoA is an important molecule in metabolism, used in many biochemical reactions. Its main function is to convey the carbon atoms within the acetyl group to the citric acid cycle or Krebs cycle to be oxidized for energy production. Citric acid cycle. an alternative name for the Krebs cycle. Glycolysis, the cellular degradation of the simple sugar glucose to yield pyruvic acid and ATP as an energy source.
Bawat bata may tanong,
Ba't ganito? Ba't ganon?
hayaang buksan ang isipan.
Sa science o agham, tayo na sa siniskwela. Tuklasin natin ang siyensya.